good, all of you. Almost all of you are here. Middle quadrant is completely gone. I don't know what happened there. Okay, so without any further ado, let me delve into what I want to talk to you about today. But before that, I want to ask all of you a question. How are you? All fine, right? Well, some of you are staring at me like that. Nobody asked you this question about. All good, right? Okay, hope you are doing well. Okay, but that's not what I want to ask you. What I want to ask you is, why? Why are you doing well? I don't mean to be rude, okay? I'm just asking why in general. Some people ask you, how are you? You say you're fine. But then have you ever wondered why? Why am I doing fine? Okay? Now, there could be many reasons why you could be doing fine. There could be many reasons why you could be doing fine. Okay? You're doing well. Many reasons are there. However, one thing I can say for all of you in common is that you have food. Okay? You have proper food. Especially if you're a day scholar, okay? So you have proper food. Yeah. So when you have food, what happens? You get nutrients from that food. Those nutrients contribute to you being well. Alright? Okay? And so you obtain nutrients. Now, I want to ask you a follow-up question based on this. Hope you understood this so far. How? I mean, you heard about it, right? 11th standard, you study this. Digestive system. It goes into your body, and then what happens? The nutrients get absorbed. Okay, so now since you're in MBBS, we are going to talk about the how, behind how those nutrients get absorbed. And that is why this beautiful mechanism called as secondary active transport. And in case you're wondering why the design looks like that, it's relevant. And it's very relevant to what secondary active transport is all about. Okay, and that is the introduction of the topic that I'm going to take for all of you. Now at a glance, these are the things that I'll be covering. I'll be covering the two major events or steps of secondary active transport, a summary of the mechanism for your benefit, the types of secondary active transport, and characteristic features of secondary active transport. One assurance I can give you right now is that if you listen to me carefully for the next few minutes, you'll be able to easily understand the textbook portion of secondary active transport. Okay, it's all up to you. I give you that assurance from my side. Okay, now let us set the stage for secondary active transport. Now what is this? This is a part of your small intestine. What is the small intestine? It's a tube, right? It's basically a tube. Tube is what? There is an inner lumen, inner central part which is hollow. Then we have the tube itself, and then the part outside the tube. Okay, that's what I've put here in a very schematic representation. We have the lumen here, that's the inner part of the tube, the epithelium, the tube itself, and interstitium, which is beyond the tube, around the tube. So these are the three regions that I've demarcated over here because this is exactly where secondary active transport is going to happen. Okay? Now, let me also talk about the contents of these regions. We have two sodium ions in the interstitium. We have one sodium ion in the epithelium and one within the lumen. Okay? They are pretty significant. You'll understand why they are there. Okay? Now let's say you eat some food. Okay? You're eating food. And then a lot of nutrients come. Let's say for the sake of example, we just demarcate one molecule of glucose. This molecule of glucose is right over there. It is near the epithelial cell. Your cell looks at that glucose molecule and your cell wants that glucose molecule. Now how is the cell going to get that glucose molecule? It doesn't have, pseudopodia that you can grab the glucose molecule just like that. It happens using physiology. So this glucose molecule is required by the cell, but the cell needs to figure out a way just to get that glucose inside itself. Okay, so how does it do it? Is what we're going to look at. First things first, you know about the sodium-potassium pump, right? We then talked about it in brief. Okay, so if you know what sodium-potassium pump is, you'll be able to understand this part. Now the sodium-potassium pump, using this, the cell pumps out this one sodium ion that it had inside of it, outside into the interstitium. I will see that. Okay, it just pumps it out. And how does it pump it out? There's more sodium ions on the outside, less on the inside, so therefore it's a form of active transport, right? And now, using active transport, it removes that sodium ion from within itself and puts it into the interstitium. Now how does this happen? It happens through the help of ATP, which is the energy. And why is it even called primary active transport? You can just call it active transport, right? The reason why it's called primary is because it directly gets its energy from the adenosine triphosphate molecule over there. It takes that energy, pumps out the sodium directly out from the cell into the interstitium. Okay? From a region of lower concentration of sodium ions to a region of higher concentration of sodium ions. Okay? So first of all, now before I go into the what happens next, okay, and such an amazing thing happens, let me tell you an analogy, okay, regarding what happens. Now let's take a ball. You have a ball with you. You throw the ball up in the air, and then the ball reaches maximum height, falls back down, okay. Now when you throw the ball up in the air, you're using what energy? Your own muscular energy. But when it reaches the air, are you using your muscular energy? No. Nature does that for you, right? Just like that, when the cell uses its own energy to push sodium ions outside. At that moment, I want you to pay attention to the lumen and the epithelium here. So what's going on? There's less sodium inside the epithelium and there's more sodium on the lumen. So what happens? A gradient is now set up. Okay? So this sodium ion now uses passive transport, which Muzaffar covered, and tries to get inside the cell. Okay? It tries to get inside, and it happens as it is. Okay, you gave the initial energy, and now nature is doing its work for you by putting the sodium ion inside. Okay? But here's something beautiful that happens. Along with the sodium ion, glucose also goes inside. Like, what's up with that? I mean, does glucose have some sort of a connection with sodium ion that it just drags the glucose in? No, right? This also happens through physiology only. And there is a carrier protein that takes care of this. It's called SGLT, sodium glucose co-transporter. Okay? Now, before I tell you the mechanism as to how sodium goes inside, and because sodium goes inside, glucose also follows the sodium, let me give you an illustration so you can understand this next part properly. Okay? Let's assume, okay, an example, you're just in your room, okay? You're just enjoying, you're being happy in your room, okay? You're just minding your own business. Outside of your room, there is a cockroach on the door, waiting for you to do something so you can come inside your room. Simple as that, okay? Now think about this. Your friend is outside the room, your friend's coming in, he's going to visit you, but instead of saying, instead of knocking your door like a regular person, your friend decides to knock down your door, okay? Your friend decides to kick the door, okay? And then he gives power one nice kick to the door. The door is sent flying and you're just sitting there wondering why is the door flying towards you, and you're just bewildered, okay? So that's happening. Now, at that instance, what happens? The cockroach looks at the door flying away and then takes an opportunity to enter inside, just like that. So why did I give this example to you? Okay, it's because of this. Your friend is the sodium ion, the cockroach is the glucose, your door is the carrier protein, and if you notice, the carrier protein has changed. So, you know that the sodium is getting inside the cell by diffusion, which means it has energy. And if it has energy, that energy is used to change the conformation of the protein. It uses that energy to change the conformation of the protein, and with the change of conformation of the protein, that glucose molecule that is attached to the protein, right, it comes inside the molecule with the help of that. So if you look at this carefully, in any ordinary situation, glucose won't be able to enter inside. There's more glucose on the inside, less on the outside, you need energy for that to happen. But now, instead of ATP, the energy is now given by the conformational change of the protein. Now that takes glucose inside along with the sodium ion. And just like that, your cell gets glucose and it keeps doing it regularly as and when required. Beautiful, isn't it? Amazing mechanism. To summarize, secondary active transport depends on indirect energy source. What is this thing called indirect energy source? Simple example, let's say you have a ball, you slam that ball and then you give some energy to that ball. Now what happens? Someone is standing over there, some stranger, the ball hits the stranger, okay, with the energy that the ball has, okay. You gave your energy to the ball, that's a direct source of energy. But because of that, the ball hit your friend, that's the indirect source of energy. But whose energy is it at the end of the day? Yours, right? It's just that it's been indirectly used to make the mechanism happen, that's all. So this is a mechanism that depends on indirect source. And what is this indirect source? It's usually from the sodium ions. Primary active transport, like you saw, ATP being directly used in order to pump this mechanism, and then the sodium ion that comes out of it, you just saw it right now, uses that chemical energy to bring secondary active transport to reality. So that's what happens, and it's usually done by sodium ions. That's what has been noticed in common. And if you want to summarize the mechanism You just need to say that sodium ion is pushed out of the cell against its gradient by primary active transport, so that you can induce passive transport of the sodium into the cell, which in turn induces passage of glucose into the cell against its electrochemical gradient. Okay, no matter what. So in other words, irrespective of the situation, you will be able to get glucose into your system because of this beautiful mechanism called secondary active transport. Okay, and with regard to the types of secondary active transport, right now we saw symport. What is symport? Glucose is going in one direction and sodium also goes in the same direction. So therefore, since it's in the same direction, it's symport. If there's symport, there's antiport as well. Sodium is going in the same direction, the target molecule is going in the opposite direction. Now I'm just going to run through the examples of these types. Symport happens in proximal conjugate tubule for reabsorption of glucose. This is how it happens. It also happens in the GI tract between sodium ions and amino acid transports. It also happens within the same mechanism also happens within the renal tubule as well. So I hope you can see this properly. And with regards to antiport, it happens on your RBC membrane with chloride and bicarbonates transferred on a regular basis. And of course in your renal tubules with sodium and hydrogen ion transport happening over there on a regular basis, along with the glucose reabsorption. Now with regards to the characteristic features of secondary active transport, which is the last topic that we'll be looking at, there are two features: competitive inhibition as well as saturation kinetics. Okay, so I'll also use analysis for this. Now competitive inhibition, let's say you got chicken, okay? You're in a hostel, you got chicken. The parents cook chicken for you, you got it, and you put chicken over there in your room, okay? And now your friends saw it. You invited your friends. Your friends are now coming inside. They want to get a piece of that chicken. They want to enjoy it, right? And now at that instance, you're also feeling hungry. However, because your friends are crowded so much around the chicken, you're not able to get that, okay? So imagine struggling and fighting to get your own piece of chicken. That's how it is. Competitive inhibition is just like that. Imagine in a molecule, in the sodium glucose transport molecule, glucose is not just competing with itself, it's competing with many other molecules to enter inside. The spaces are limited, and even those limited spaces where reactions happen, glucose wants to enter, and glucose is competing or fighting against other molecules just to enter inside the cell. So that's competitive inhibition. And with respect to saturation kinetics, we can use a follow-up of the same example I used. You are having this chicken, and then your friends notice it. They are coming from the outside. They notice it, and all of them get so excited, they try to enter at the same time. What happens? Nobody enters. They're all stuck at the door itself. Nothing is happening. Just like that in saturation kinetics, in the protein, right, there are only very limited number of active sites where the reactions happen. And because the number of reaction sites are limited, the number of molecules that could attach to the protein molecule are also limited. And because they are limited, obviously not all glucose molecules get treated properly. And just like that, that's why after a certain amount of concentration, it reaches saturation. So that's why one limitation of secondary active transport is saturation kinetics. So these are the two features: competitive inhibition and saturation kinetics. With that, I would like to end my presentation by saying the references. I refer to page 45 of Leiden and Hall and page 39 of GK Paul. If you understood everything so far, you'll be able to read these pages easily, okay? With that, I will thank you for the opportunity and thank you very much for your time.